Welcome to The Explainer. Today we're going to do a proper Journal Club deep dive. We're tackling a fascinating and, let's be honest, methodologically dense paper from Wei and colleagues, published in 2023. This paper really tries to finally settle a long-standing debate. What is the actual functional legacy of Neanderthal DNA in us, modern humans? So to really get what this paper accomplishes, we're first going to set the stage, trace the scientific back and forth that led us here, then we'll dig into the big analytical problem they had to solve, see how their new method works, and, of course, look at their major findings. We'll wrap up by looking at the specific Neanderthal genes they pinpointed and the big questions that are still on the table.
Okay, first things first. To really appreciate what Wei and his team did, we need to understand the question that's been bugging this field for more than a decade. What are the real, measurable effects of our Neanderthal DNA on our traits today?
Right, so ever since Green and colleagues dropped the draft Neanderthal genome on us back in 2010, it's been a bit of a roller coaster. Pretty quickly, in 2014, Sankaraman and others suggested that a lot of these Neanderthal variants were actually being selected against. But then you had studies like Dannemann and Kelso in 2017 finding these really cool associations. Then, just as things were getting interesting, Scove and his team in 2020 came along and basically threw cold water on a lot of it, arguing that most of these were just statistical ghosts. And to make it all even more confusing, MacArthur and colleagues found evidence for elevated heritability in 2021. So, you can see the problem. The field was kind of stuck.
And this quote from Wei and colleagues really nails the problem. The core issue, as they put it, is that these introgressed variants, the bits of DNA from Neanderthals, are just, well, they're weird. They have their own unique history, and that gives them statistical quirks that completely mess with the standard tools we use for modern human DNA. So what are these idiosyncratic properties exactly? What are these statistical gremlins that have been causing so much chaos and contradiction in the research? Let's break it down.
First off, the authors had to define what they were even looking for. They honed in on just over 235,000 specific genetic variants that are really good signposts for Neanderthal ancestry. They gave them a name, Neanderthal Informative Mutations, or NIMS. So whenever we say NIMS, this is the specific set of variants we're talking about.
Okay, this slide is the absolute crux of the analytical problem. Compared to your run-of-the-mill modern human variants, these Neanderthal NIMS are different in two really important ways. First, they're usually rare. That's a lower minor allele frequency, or MAF. And second, they tend to be inherited in these big, sticky, correlated blocks, which means they have much higher linkage disequilibrium, or LD.
In this chart, which is inspired by Figure 2 in the paper, just shows you how dramatic one of these differences is. That average LD score, which is a measure of that blockiness, is way, way higher for the Neanderthal variants. I mean, look at that. It's more than 2.5 times higher on average. These aren't just tiny little statistical quirks. These are fundamental structural differences in the data.
So here's the big takeaway from all this. If your analytical model doesn't specifically account for both of these properties, the frequency and that blocky LD structure, your results are going to be biased. It's almost guaranteed. And Wei and his colleagues argue this is exactly what happened, and it's the root cause of all those conflicting papers we saw on the timeline.
Okay, so how did Wei and colleagues fix this? How did they manage to build a better mousetrap, so to speak? Let's get into the methodological guts of this paper.
Their solution is actually really elegant. They start with a very powerful statistical method called RHMC, which is built to handle huge datasets like the UK Biobank. But the real secret sauce is their annotation strategy. They slice up the genome and then build a model that controls for everything at once. Is it a SNP or not? What's its frequency? And what's its LD score? They account for all three simultaneously. And they didn't just assume this would work. They proved it. They ran a ton of simulations, which you can see in their Figure 1, and they show that their full Ancestry plus Math plus LD model was the only one that spit out accurate, unbiased results. Any model that left out MAF for LD just failed. It was the smoking gun they needed to prove that these factors were messing up previous studies.
All right, so now they've got this new, super-calibrated method, the moment of truth. They could finally ask for what they hoped would be a clear answer on the overall impact of Neanderthal DNA on our traits.
And here it is, the headline finding.
Once you do a proper apples-to-apples comparison, pitting a Neanderthal variant against a modern human one with the exact same frequency and LD profile, the Neanderthal one contributes, on average, 59% less to the variation in a trait. That's not a subtle difference. That is a massive, massive depletion.
So, let's break that down a bit. Across 96 different human traits, they found that 17 of them showed a statistically significant depletion of heritability coming from Neanderthal variants. And here's the kicker, and this really sets the record straight. Not a single trait showed any kind of enrichment. Zero. This whole pattern is a huge boost for that long-standing hypothesis from researchers like Jurek and others, that for the most part, natural selection has been actively weeding out Neanderthal DNA from our gene pool.
But hold on. An overall story of depletion doesn't mean that every single Neanderthal variant is useless. The next big challenge was to sift through the noise and find the specific NIMs that are actually doing something, the ones that are truly causal. To avoid that trap that Scove and his team warned about in 2020, where a NIM looks important just because it's physically close to the real causal variant, they built this incredibly rigorous pipeline. They start with a super strict significance threshold. Then they use a fancy Bayesian tool called SUSY to really zoom in and dissect the genetic region. Finally, they apply these tough filters to make sure they only keep the hits where the NIM itself is the most likely culprit.
And you can see right here just how essential that whole pipeline is. Their own tests showed that if you just did standard association testing, you'd have a false discovery rate of almost 30%. Nearly a third of your hits would be wrong. But with their full pipeline, they slashed that error rate almost in half, which gives you way more confidence in the variants that make it through.
So what treasures did this pipeline uncover? Well, they ended up with 112 genomic regions that contain what they call credible NIMs. And when you look at the genes in these regions, it's fascinating. You see a bunch involved in the immune system, like FCGR2A and STAT2, which, by the way, was one of the very first candidates for adaptive introgression ever proposed. But you also see genes involved in metabolism, like AKR1C4's effect on bilirubin, and even development, like COQ10A's connection to standing height.
So, let's pull back for the big picture. What's the main takeaway from this study? And where does this leave this really exciting field of research?
Look, the number one impact of this paper is methodological. It gives the whole field a much more solid, calibrated toolkit. But the authors are really clear about what the study doesn't do. For one, it's all based on one population, so the story could be different in other parts of the world. And while they found this overall depletion, they can't quite tell us yet if that means there are fewer Neanderthal variants having an effect or if their effects are just smaller. Also, that pipeline is so tough that it's probably being very conservative, meaning there are likely a lot more functional NIMs out there left to find.
And for any researchers watching who want to go deeper, the bibliography of the Way paper itself points to these as pretty much essential reading. These papers really lay the groundwork, covering the core concepts of purifying selection, adaptive introgression, and the whole genomic landscape of our Neanderthal ancestry. They provide the critical context for this new work.
And that really brings us to the final, and I think most tantalizing question that this work leaves us with. If the main story is that selection has been systematically getting rid of Neanderthal DNA, why did some of it stick around? Specifically, why the bits that are involved in things like immunity and metabolism, our interface with the outside world? What was the evolutionary tug-of-war that made these specific pieces of our archaic past valuable enough to keep? That's the complex puzzle that the next wave of research, building on this fantastic paper, is going to have to solve.